Well, good day to everyone joining us and welcome to today's X-Talks webinar. Today's talk is entitled AI in Bioinformatics, Overcoming Pitfalls in Statistical, ML, and Generative AI Approaches. My name is Ryan Muse and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speaker. Now, the webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and your comments for our speaker throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, you can contact me at any time by sending a message using the same chat panel. I'd also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you to submit your questions as well via the comments tab. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming to those who registered on xtalks.com. At this point, I'd like to thank BullFrog AI, who developed the content for this presentation. BullFrog AI leverages artificial intelligence and machine learning to advance drug discovery and development. Through collaborations with leading research institutions, BullFrog AI uses casual AI in combination with its proprietary BF-Leap platform to analyze complex biological data, aiming to streamline therapeutics development and reduce failure rates in clinical trials. Now I'd like to introduce our speaker for today's event. Dr. Juan Felipe Beltrán is an accomplished scientist and software engineer working in algorithm development, ML, and bioinformatics. Dr. Beltrán has a proven track record in designing and implementing innovative solutions for complex biological data analysis, advancing proteomics genomics research, and contributing to the fight against global health challenges like COVID-19. But now, without further ado, I'd like to hand the presentation over to our speaker, Dr. Juan Felipe Beltrán. You may begin when you're ready.
Thank you, Ryan. Good morning, folks. We're going to be going over 3 separate pitfalls today that are common problems I find when I'm working in bioinformatic analyses that are trying to find a place to live between using rigorous methods, interpreting machine learning models properly, and applying the new tools that we're finding coming from generative AI. So this will be structured like 3 micro talks that come together towards a way of thinking about the data that is so challenging to deal with when you're working in biology. As far as my background goes, one of the things that I like noting, especially for this type of talk, is I started in computer science and moved towards computational biology for my PhD, motivated by finding increasingly more difficult data sets to interact with, and I got more than I bargained for in biology. So my favorite example will be our first area, mismanaging compositional data analysis. So compositional data is a term that might not be as common as it should be in when you're talking about analysis, given how common this type of data is. Compositional data is any type of data where you're looking at a fixed whole that is divided among parts. This can be the percentages of a budget, this can be the grams in a recipe, or this can be the reads in a microbiome that you're trying to analyze. The thing that makes compositional data compositional is that no individual piece of information, no individual measurement gives you the whole story unless you know what the other measurements were. So to build some intuition around this, I'm not going to pick budgets or the microbiome, as those tend to get a little unwieldy and unintuitive. And instead, I'm going to focus on grams in a recipe as a way to build intuition for the pitfalls that we can encounter here. So to start, we're going to think about a simple recipe for sugar cookies. You're going to be mixing some grams of sugar, flour, and butter together to make different batches of different sizes. And here you can see that we've kept sugar stable. Every single batch is going to be using 25 grams of sugar. You can imagine as you make these batches, you can taste them, you can rate them based on sweetness, which is going to be our last column on the right. And it should be trivial, you would think, to build a model that is able to tell you that sugar is sweet. That should not be a challenging thing to find. But if you're using naive analysis, you quickly run into the first problem with compositional data analysis. If sugar is stable, but flour is increasing every time, you might just find that adding flour makes things less sweet. You might find that butter seems to be moving randomly and it's not contributing at all to the sweetness score. but sugar is not going to be contributing either if you're using naive analysis. If you're doing compositional analysis, you're trying to think about it as what percent of the recipe is sugar? What ratio of sugar to flour are you looking at? And I want to talk a little bit about the types of decisions that you make about how you represent this data in order to do this. But first, I want to complicate it a little bit more so we can get closer to the biological case rather than the simple case. So one jump in complexity here would be moving from a sugar cookie recipe to multiple recipes that share some subset of ingredients. Now, if you're trying to rate sweetness, things are a little more complicated. You know that there's two different types of sweeteners now. We have sugar and honey. And we know that in the ground truth, sweetness is going to be some weighted combination of the amount of sugar and the amount of honey relative to the amount of other ingredients. And here you start seeing some of the hiccups that are going to make computational analysis difficult in these types of data sets. First, if you have any recipe in your data set that has 0 flour, butter, and cocoa, you're dividing by 0. So you need to make sure that you have a way to avoid this. Any bioinformatician in the audience might both have in their pocket the suggestion of adding a pseudogram or a fake gram of any ingredient that is 0 to avoid zeros. And anyone who's been cooking starts feeling the intuition for how odd it is to add a gram of an ingredient that is not part of a recipe and how much that might lead to misinterpreting what the recipe actually is. There's a lot of argument about how you solve this problem, this type of problem, but again, I want to reach as close to biological complexity as we can before we start talking about methods. So sampling is the last thing we're going to introduce here. Here, I'm showing you 4 different recipes from which we've taken 3 samples each of 10 grams. And this is where compositional data really starts getting complicated. We're no longer looking at the full ratio of the original recipe. We're going to be having some variance depending on random chance. How much sugar you get from a sugar cookie bowl, if you take a tablespoon, is not going to be consistent every time. Here, if we start thinking about the most trivial compositional transforms, like turning all of these into percentages, we start seeing the types of systematic problems in analysis that are going to start popping up. The first one here on the right is our within recipe variance explodes. If we're looking at sugar cookies, sugar percentage, we have 30%, 20%, and 40% of the recipe with a standard deviation of 10%. We're having a lot of uncertainty about how much sugar is actually in this recipe based on a percent transformation alone. And this leads to false negatives. If you try to run a statistical test to differentiate between sponge and sugar cookie, it's very easy at the recipe level, but it's going to be very difficult at the sample level due to this heightened variance. Finally, just based on the sampling and the compositional nature of the data, you're going to have spurious anti-correlations. This is something that Fisher, one of the fathers of statistics, warned about ominously the few times that he talked about compositional data. There's the problem of spurious correlation. In this case, sugar and flour percentage will appear to be perfectly negatively correlated in every bowl, even though that's not the case. That's not what's happening. So we established that we can't use the raw counts or the raw grams in a compositional analysis, at least not without a very smart model. And percentages are not quite enough. So how do we talk about what the right transformation might be here? How do we avoid these types of pitfalls? This is a pretty fantastic paper that I strongly recommend from Lior Pachter's group, Depth Normalization of Single Cell Genomics Count Data. They take 526 single cell experiments and look at the way in which single cell transforms affect their data. And to highlight the bottom lines, I've adapted some of these panels to make them a little more readable. Here, we're measuring the coefficient of variation. The higher, the worse it is. So you don't want to have high variance in your data set that is artificial. On the x-axis, we have the different ways in which you could represent your counts data. Raw is going to be just the counts. So this is going to be recounts. In the recipe case, this would be the number of grams. We have PF, which is percent fraction. This is a simple percentage. And then we have 5 transformations that are trying to shrink variance explicitly, taking the square root of the counts, taking the log 1p of any of these measurements, and one extra feature that I'll discuss, one extra transformation, PF log 1 PF. Now, to understand what's happening with these orange features, I just wanted to highlight log 1 PF is simply taking the log of a feature with a +1. This can be your added gram to avoid taking the log of 0. And you apply it to either raw, counts per 10,000, counts per million, or the percentage from before. PF log 1 PF is an exciting transformation. I'll show you why before I tell you what it is. Here we can see most of these shrink variance. That is good. But there is another component that we need to look at, which is the correlation with cell depth. This basically comes down to how much your data is just representing what the total amount of sample that you took was. So if you took 20 grams versus 10 grams, all your features are going to be inflated in the 20-gram case. This is trivially true in the raw case. If you're looking at the counts, that's going to be 100% correlation with how big your sample was. If you take a percentage, it's going to be 0% correlation. All the features that looked really good in the previous plot have really high correlation with cell depth, but PF log 1 PF does not. And this makes it a really attractive heuristic, a really attractive choice if you have a compositional data set and you want to start examining it closely. PF log 1 PF is simply take the percentage of your feature, apply the log with this added +1. Funnily enough, this takes 0% to 100% and 100% to 200%, and then take the percentage of that. It's a little unintuitive, but it keeps your variance low while also destroying that correlation with cell depth that's going to cost you when you're trying to identify what are the actual drivers or what are the actual important genes in a particular data set. Now, this is not a silver bullet. You usually want to pick your transformation based on the method that you're using. We use different methods of BullFrog AI to analyze count data. Some of those methods assume distributions that work best if you give them the raw counts. They know that these are raw counts from some composition. Some of them are Bayesian methods or simpler methods that do benefit from something like a PF log 1 PF transformation. So this first pitfall is where I see a lot of teams struggle. How do you transform the features, and why is it important? What are you trying to avoid? I hope that, especially with simplified recipe example, you can start getting the intuition that yes, you will get results. You will get a result that is not technically incorrect, like the more flour you add, the less sweet a dish is going to be. But it's not going to be capturing the actual drivers of the behavior if you're not analyzing your data properly. Sugar being unrelated to sweetness is trivially not the correct result in these types of situations. Now, for pitfall number 2, we move further down in the pipeline. Once you have a data set that you have transformed and you're trying to draw inferences from, how do you decide which features, which genes, which ingredients are contributing more to a particular decision? Now, in a typical tabular machine learning setup, you have some number of samples and you have some number of features describing those samples. And you have a special type of feature, your label, that is going to be 1 or 0. It's going to say this came from a cancer cell, or this came from a control cell, this came from a rainy weather sample or a sunny weather sample, a simple binary. You build a machine learning model that's going to try to predict these labels based on the data. And usually that can be applied by itself. You can save that model and you can apply it to new problems. But there's a secondary output from training a machine learning model that's very useful. And that is a measure of which features are most useful to learn the label. Depending on what type of model you're using, this measure is going to be something different. If you're using regression models, this is usually going to be about the magnitude of the coefficients. How large is the number, the weight that you're multiplying your feature by to get close to the answer. If you're doing tree models, it's going to be about looking at every time a feature was used to split a data set and how much that split increased the performance of the model or how much it decreased the loss function. In neural networks, it's going to be more complicated. You're going to be looking at something like the amount of the gradient that is tied to deviations in that feature. Or if you're being really practical about it, you take the feature, you shuffle it so that it's in the wrong order relative to the rest of the data set, and you see how much the performance of your model drops. And you do this over and over and over to get a measurement of how important that feature is. Regardless of what feature importance measure you're using, I'm a big fan of trees. There is one thing that often gets skipped that I think causes a lot of damage in analysis, and that is running the experiment more than once. Feature importance, like anything else, is taking a measurement, trying to calculate an underlying truth value from a subset of data. In this plot, what I'm showing you is what happens when you take 80% of a data set with the same labels and you just train it over and over and over, and you start aggregating the importance measurement for features across 10,000 models that you've built that technically have the same exact task, just with slightly different subsets of the data. Here I'm highlighting the 10 most important features and the 10 least important ones. You can see that importance is eventually stabilized, and that is good. We want them to be stable. But if you've only trained one model, notice how unstable even the ranking of features is early on. There's a point at which even our least important features start being competitive with our most important features early on in the process. This is noise that needs to be taken into account by sampling. So our first insight that we want to share when talking about measuring feature importance is you do need to run multiple trials, and your feature importances should stabilize. One less intuitive insight is what happens when you're not using real labels. And this feels like an odd thing to do, but bear with me here. Let's say that instead of having some real labels corresponding to how many of these samples are cancer cells and how many are not, instead you have some probability of this being a 1, and you set it for everyone at 0.4. You can use this to generate different iterations of random labels that you'd have. On average, 40% of them should be 1s. Each of these iterations is unconnected, is not connected to any of the others. When you run the same process of 10,000 models, these feature importances, though they're more chaotic, they do converge as well. And this should be surprising. Basically, what we're seeing here is that some features just have a baseline feature importance that you're going to get no matter what. Why does this happen? It's very easy to think about this if we oversimplify it. So I'll start there, just like recipes tend to be a good way to oversimplify compositional data. So consider 8 samples with 2 features. Feature 1 has 1 0, everything else is a 1. Feature 2 is half zeros, half 1s. If a single feature is being used by a machine learning classifier, we know that we could turn this into a 1 and all of these into zeros, or we could turn these into 1s and these into zeros. But we could never split the 1s. We could never separate the samples that have the same value in a feature using that feature only. These are identical as far as that feature is concerned. So here are 4 examples of labels that we're going to try to classify as best as we can with either of these 2 features. The number on top shows you for feature 1, 3 have to be incorrect. This is the most correct that you could get the prediction with feature 1. This is the most correct that you could get the prediction with feature 2. And there's a trend that starts appearing if you do this across all possible counts of how many 1s we have in that label, that will show you that feature 2 will be expected to have more errors when you have few numbers of 1s or few numbers of zeros, but feature 1 is expected to perform better in the middle. This type of phenomenon is feature importance that is biased by the number of unique values, by the cardinality of a feature. It's a toy example, but it captures a phenomenon that keeps appearing when you use more complex features. An analog of this comes out when you allow features to work together. Suddenly, you could have 1 type of data point, 2 types of data points, 3 types of data points, based on combinations between feature 1 and feature 2. Alternatively, once you move into a continuous space, you can have features that are correlated, collinear, or orthogonal. And they might be orthogonal with other features. There's some baseline level of relationship of joint complexity that makes some features useful in general at classifying certain types of labels, certain populations of labels. So when you're trying to measure feature importance, the big insight here is you shouldn't just run one model. You should run a ton of models to get a good estimate on your feature importance. And you should run a null models that are trying to classify something that looks like your data but isn't quite your data, and combine them to get a normalized feature importance. This is a process that we've tuned quite a bit at BullFrog AI. And we see that it allows us to combine information across classifiers, across data sets, in ways that really maximize our ability to use as much data as possible for making decisions. The last pitfall that I wanted to touch on goes into the biggest hype that we have in tech right now, AI models and how to use them in a smart way. I want to show you a prompt that may seem like it is a great fit for a language model and is actually pretty bad. Order the following one-sentence research paper titles by the likelihood that the finding will replicate. And we give it a list of paper titles. Now, there's many different ways to think about how large language models work, or how do these agents, AI agents work. For this presentation, I'd like you to think about it in terms of how many minutes of human effort you think it would take to complete this task. If you put yourself in the position of the model answering this question, ranking all of these becomes very difficult, not just because estimating whether a finding will replicate is fuzzy and kind of a judgment call, but also because anything that requires sorting requires some amount of comparison between different items in the list. Now, why do I want you to think about how long it would take a human to do this? As we've been releasing models over time, we're seeing that they're able to complete tasks that take humans an increasing amount of time successfully. But this is not 100% success. This is at 50% success rate. And when you look at this plot, you might start thinking about the other side of this question. Does that mean that the longer it takes a human to complete a task, the higher the error rate is for a language model? And the answer is absolutely yes. If you look at analysis done by external organizations, by Anthropic, or by OpenAI, we see that across the board, human time to complete is a really useful predictor for how often the models will be able to complete that task successfully. And this doesn't just cover our normal LLM models like GPT-4o or Claude Sonnet. It also covers agent models like O3, where they're able to take many different iterations to think or to take many steps. It's not about the amount of time that they can spend on it. It really is a bound on how much complexity can these models handle at a time. Now, how do we use this to avoid the pitfall of giving it a huge list and hoping that it can sort it? Here, the typical risk is it will give you some output. It will always answer the question, but will it replicate? Is it correct? Does this fall in the 50% success or 50% failure case? And how do you double check it? Now, kind of echoing everything else about my approach during this talk, the real answer is to treat this as a measurement device, to realize that the LLM is not solving the entire problem, but it is part of an instrument that you can use to answer questions. So instead of asking it to rank all of your options, you can take your options, pick two of them, whether it's based on random picking, a tournament, or a smarter approach. You just need to pick two of them at a time and ask a model to judge which of the following research paper titles is more likely to replicate, A or B. This is a competition. The model will tell you whether it believes that in this competition A or B won, and you can use that to update some skill ranking model. If you're into chess, this would be equivalent of an ELO rating for each of your candidates. How often do they win in competitions against other models that other candidates that also have their own skill rating? And what's interesting about this type of framework is it's built to handle cases where the judge will not be consistent, where sometimes B beats A, even though before A beat B. There's many ways to extend these models because we've been looking at competition for a long time, and competition doesn't always give you the same outcome. It uses the fact that the large language model is not going to be perfectly consistent, but that there are trends in general that we want to capture and quantify. One very interesting application of this that we find in biology is finding genes based on particular annotations. Now, we ran an experiment where we used this type of ranking algorithm to rank the relative viability of two proteins as targets for type 2 diabetes out of a set of 250 random genes, plus a spike-in, which had all the genes that were annotated with type 2 diabetes, CRC, and obesity in the UniProt database via their disease column that comes from OMIM. After ranking, I want to show you the top 10 targets because they paint an interesting picture of what we're able to do with these language models when we break down the task into small pieces. In light blue, we have our spike-in. These are the genes that were annotated as being associated with type 2 diabetes in UniProt already via OMIM. Insulin receptor, GLUT4, it's what we expected to find. In dark blue, we have something that should be very familiar to bioinformaticians. These are targets that are actually annotated for type 2 diabetes in the original database that UniProt pulls from, but as is often the case in biodatabases, some identifier matching or some filter kept it from making it all the way to the feeder database that we were looking at. So these two were recovered as technically cases where they should be light blue. In orange, we start getting some of the exciting cases. These are not annotated as being associated with type 2 diabetes in OMIM, but they are in co-therapy trials with GLP-1 for type 2 diabetes and obesity. And arguably, these are viable targets for type 2 diabetes. Now, why don't they show up in annotation? It comes down to specificity. OMIM is specifically about Mendelian inheritance. It's about disease where we can see the way in which it is inherited based on variants in these genes. And it's a level of specificity that's not what we're usually looking for when we're running other types of searches for gene associations. But across the board, we're going to find that databases have their own niche that they're studying, their own definition of what association is. And these types of ranking models allow you to build your own annotations on the fly for anything as vague or specific as you need it to be. Echoing how ridiculous it might be to run a model and decide that sugar is not correlated to sweetness, here we see that no database annotation would have made it straightforward to fish out these purple cases, canonical regulators of glucose metabolism, that are definitely associated with type 2 diabetes. They are interesting targets for type 2 diabetes, but they would not come up in database searches as easily because they require a little bit more domain knowledge to associate them. They're not going to be associated directly with the term type 2 diabetes any more than you would find genetic evidence for glucose and type 2 diabetes being related, even though they are. It's just an odd question to ask from the databases that are available to us. So this covers three very different types of areas where it really comes down to simplifying the problem, making sure you know what it is that you're measuring, and building a reasonable model around it, whether it is looking at your compositional data and understanding what are the types of artifacts that are going to be created in the data. It's going to give you an answer, but you need to make sure that it is correct. In pitfall number 2, we see this again with machine learning. You can train a machine learning model. You can ask for feature importance, and it will give you an answer, but you need a model to make sure that this answer is correct. And again, echoed in number 3, you can ask a large language model to solve any problem for you. But without building a model, you will just take the answer that it gives you, and you will not know if that answer is correct, have error bars around it, or understand how much it would replicate. Across the board, our thesis ends up being treat all of these data generating sources as measurements with their own noise, with their own quirks, and with the necessity to build models around them that allow you to extract the most useful information from them so that you can make decisions without as much fog of war as you would do otherwise. We use this across the board in our analytics stack at BullFrog AI, where we've built many modules for feature importance, measurements, similarity and clustering, structure discovery, and lead annotation and ranking. A lot of this is built around a very exciting code base that we got exclusive access to from the APL, the Applied Physics Lab at Johns Hopkins, that is incredibly apt at graph analytics. It's very useful when you're trying to describe complex relationships in a complex network. We use this to generate different types of outputs, both internally and for our clients, like sample-to-sample similarity networks and clusters, or feature-to-feature networks that show you the dependence of different features. In this case, we're showing you a version of this that represents a gene causal network. These are very useful for tasks like novel target discovery or finding patient subtypes for clinical trial optimization. We've been very happy with the types of results that we're able to extract by making sure that everything we're feeding into the APL models and everything we're processing out of the APL models understands the underlying statistics, understands the underlying measurements, and measures the error throughout so that we know when we're wrong instead of just taking any answer as the answer. If this resonates with you in any area, whether it is problems where you need to have a way of judging whether it is correct or not, data sets in biology where you need patient subtyping or feature dependence, causal networks to pick where to intervene or where to move upstream from a target that is overpopulated, we would love to chat with you. My email is on screen and you will receive it afterwards. And thank you for your time. Ryan, do we have any questions on the chat?
Yes, well, thank you so much for that very insightful presentation. As we move into the Q&A session, I'd like to remind the audience to keep sending their questions or comments with the questions window if you're attending via GoToWebinar. For those watching on LinkedIn Live events, you can use the comments section. Now, I've already got some questions from the audience, though, so we'll get ourselves started with those. The very first question that I have here, it just says, what tools would you recommend to conduct multi-omic analysis, or are we better off making custom tools for our specific use cases?
We host a couple of tools for multi-omic analysis, but we had to build them from scratch because across the board, I would say that there's no easy solution. If you want to build EQTLs, there's a couple of modules that will let you do that for that specific pair of omics data. But for true multi-omics data, it comes down to what is the question that you're asking, and no single library will be able to support that. Usually, we approach this by using that main pipeline, but inevitably, we got to focus on the question. So we look at the layer at which you're asking your question. Is it a subtyping question in multi-omics? Is it about predicting one of the omics outputs from two of the others? Overall, I have found that more time has been wasted by trusting an existing library than by building some of the analysis yourself or making sure that you go down into the underlying code of the existing tools and verify that it's doing what you think it's doing.
Perfect. Thank you so much. Now, I've got another question here, and I think you spoke to this a little bit at the very end. Someone's wondering if the AI technology used in your presentation is the JHU Applied Physics Labs breakthrough AI technology.
I think some of it falls under that umbrella, but I'm not familiar enough with that term to tell. This is the Applied Physics Labs libraries that, if you're looking for news releases around them, would have the names Euclid, Siegel, or Socrates coming out of APL.
Perfect. Thank you. That was very helpful, I'm sure. The next question here is someone curious. They see BullFrog AI has an R&D pipeline. How are you using this technology to advance those programs?
So internally, we run our own CNS R&D programs where we have our own candidates that we're trying to advance. We use them for identifying subsets and heterogeneous disease data sets, and then identifying targets for those subsets. Additionally, we also work with external clients to do this type of analysis for their data and towards their purposes, whether it's target identification or clinical trial optimization, where you're trying to not find a target as much as find a way to measure how patients are different and will respond differently to therapy.
Okay, wonderful. Thank you. Now, someone's wondering if you could just explain the feature importance issue again.
Oh, absolutely. When you're looking at a data set that has especially in biology, you'll have a lot of features, and you're trying to figure out which of these features predicts the label best. The machine learning model is seldom testing just one feature at a time. It's trying different combinations of features and seeing how it can build a model that is predictive. When you're trying to ask which of these features was most useful, you'll have to go back and look at one of the pieces of the structure that the model built. So focus on trees, because trees will make it easiest. When you build a simple decision tree on a data set, you can look at a feature and ask, when did this feature come up in the tree? And let's say it came up twice, and you look at what the split that happened based on that feature was, and how good was that split? And this will be a number like the decrease in entropy. You could take the sum of that number and say the goodness of this feature is that sum. But if you build another decision tree, that sum will oscillate a little bit based on which samples were picked, based on which features appeared before it and after it. So usually average this over many different trees. And that is the first part, the first insight in the presentation, is that this progressive averaging is important, especially with subsetting, so that you get as good a measurement, as accurate a measurement on this number as you can. The second aspect of feature importance is if you run this on noise, on a label that isn't actually the label that you're looking for. So for example, if you have 20% of your patient or of your samples are cancer cells, the other 80% are normal cells, and that's what you're trying to differentiate. If you just run the same features on any other mix of 20 to 80 ones and zeros, some of these features will still have a very high feature importance. And that is your null hypothesis. That is your baseline expectation. So if you want to know, what you really want to know is which features are more important than you would expect at random. That is the key of the feature importance section. We have a lot of algorithms to do this and to make sure that our measurements are as accurate as they can be, but that ends up being part of the bolt.
Perfect. Well, thank you so much for that answer and for all of the answers today, but we have reached the end of our time. If we couldn't get to your questions, know that the team at BullFrog AI will follow up with you, or if you have some further questions, you can direct them to the email address that's up on the screen. I want to thank everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your exit, and your participation is appreciated as it helps us to improve our webinars. Additionally, I've shared a link to view the recording of today's event in the chat box, which you can also share with your colleagues so they may register for the recording here as well. For those watching via LinkedIn Live events, we encourage you to visit xtalks.com to register for this event and then gain access to features such as viewing that recording and more. Now, please join me once more in thanking our speaker for his wonderful time here today. We hope that you found the webinar informative. Have a great day, everyone, and thank you for coming.